Next bit, we get to move into a panel discussion. I'd like to introduce Narges Bani Asadi, who is the founder of Bina Technologies, one of the groups that's really bringing engineering to bear on this problem, and I'd like to ask her to introduce the panelists.
Thank you, Matt. Hello, everyone. Good morning. So I'm Narges Bani Asadi, CEO and founder of Bina Technologies, now part of Roche Sequencing. Today, we have a very interesting panel about next-generation sequencing data and the opportunities and challenges that it is bringing forward for bioinformaticians, scientists, and clinicians in the industry. So before we start the panel, just a few words about Bina and Roche Sequencing. We are part of a larger family in Roche Sequencing since last year. The vision at Roche Sequencing is to create a very comprehensive end-to-end sample-to-answer workflow for genomics. And in the last couple of years, we have made a tremendous progress around different technologies for sample preparation, capture, DNA isolation, all the way to analysis and informatics. And the role that Bina plays in Roche family is to build the informatics platform, analysis solutions, and interpretation of genomics information. So today we are going to discuss together what are the challenges in front of us, what are the unique problems or opportunities next-generation sequencing is creating for the industry, whether it is in research or clinical diagnostic. But we want to also hear what you think about it. We have thought about it a lot in the last few years, and we had several good conversations with our panelists that you will hear today, but we also want to see how you think about it. So we have put together a poll. If you go to bina.com/poll, you will see it's very simple, just 10 questions. These are the 10 top challenges that we have been hearing in the field from many different types of users, and we want to see how you think about them. Do you think they are real? Which one is more important? So I want to take a moment and introduce our panelists, which I think are bringing very interesting insight to this discussion. We have Dr. Rong Chen. Rong is representing clinical research and academic research of interpretation of next-generation sequencing. He is an assistant professor and director of Clinical Genome Informatics in Icahn School of Medicine in Mount Sinai now. His research is focused on using big data approach to interpret human genome for different clinical applications. Rong is well known to many of us. He has published many papers, over than 70, I believe, in translational bioinformatics in very well-known journals. He also is very active on the commercial scene. He actually was one of the early advisors of Bina, and he has also helped launching 2 companies, Personalis and LifeMap Solution, and he's also advising to Genomics. Our other panelist, Dr. Matt Brauer, he is representing the pharmaceutical and drug discovery applications actually of next-generation sequencing. He's currently leading the Genomics team at Genentech and handling all the high-throughput data for research and development sciences at Genentech. His education is in statistics and biology and biochemistry at Berkeley and UT Austin. And he also spent some good amount of time at Stanford and Princeton doing his postdoctorate studies. So thanks so much for joining us in this panel today. So let's talk about the challenges. We spent a lot of time to think about how to organize it because there are just so many different challenges we hear about. And we came up with 4 buckets that we think might simplify the problem a little bit. So today we are going to talk about each of them. The 4 buckets of challenges are scientific challenges around making sense out of the data and link it to biology or clinical outcomes. The people challenges, which is around how we find good bioinformaticians, how we have bioinformaticians, clinicians, scientists work together in solving these complex problems. Process challenges, which is about how to govern the data, how to have flexible yet really auditable and secure, reliable processes around consuming this data in organizations. And technology challenges, of course, around how to manage this information, store them, and do the compute. So let's start to look at the scientific challenges. I wanted to ask our panelists, maybe Ron can talk to us about what are the scientific challenges he's seeing in his day job around making sense out of genomic data?
Sure. Thanks for introductions. For the accuracy side, for cancer precision medicines, we very often found somatic variants have very, very low allelic frequencies. So how we get to really even lower than 1%, 0.1%, that's one challenge, not just from the technology side, but also from algorithm side. So currently, the variant calling is always in general, but we need to really tune this algorithm to detect these low abundance variants, because often we see the resilience mutation is already there, but just in very subclone, very low frequency. And we have been found in the published data that the variant also there, but people didn't capture it because it's so low abundance. From the annotation side, I think two key. One is the population frequencies. So we recently curating 150,000 genome and from 50 different disease cohorts and so on, putting there called divas. But I think we need to do more. We really need to get to like 1 million genomes, every single variant. We know what a frequency in hundreds of thousands of disease populations. Only by that we can really understand which variants, how much contribute into the disease. And the second part is function variant. It's urgent need. We need a database with all the variants that has a functional impact. So current algorithms only saying is damaging or likely damaging. That's definitely not enough. We need to put all this functional impact into a context. Is these variants the break expression of the gene? Like, for example, currently there's not even a good database to say all the activating mutations, right? We definitely need to put this functional impact into the context, gene level, protein level, system levels, and so on. And from the interpretation side, one is for healthy subject, we still cannot predict disease risk well. We need more G by E analysis to really better risk modeling all these disease. Maybe more not in general, but let's say example are among type 2 diabetes patients, can you predict who get Alzheimer's? So these kind of specific questions have a good risk modeling.
Thank you. Very good examples of scientific challenges ahead of us. The next set of challenges are around process challenges about how to build end-to-end analytics frameworks, how to govern the data and keep it secure and private, and many more details. So I think Matt is spending a lot of his time solving these challenges at Genentech. Do you want to talk to us about it a little bit?
Yes, thanks, Narges. So just a little bit of background and context. My group is in the Bioinformatics department, which is a research department at Genentech, and historically, we've been responsible largely for preclinical data. So this is our in vitro and in vivo studies for the genomics data. And we've supported a large group of computational biologists and bioinformatics scientists. And we've really been walking the line of trying to maximize the opportunity for innovation versus keeping a lid on it to some extent and ensuring reproducibility. It's actually fairly catastrophic for us if we get different scientists coming up with different answers, and it leads to some major investment decision. From that context, we've recently started moving into a position where we're realizing that a huge amount of our assets are related to the clinical trials data that we have. So more than half of the drugs that we have in the clinic have a strong genetic component. And we know that the gene association studies, if we could look at those, the genetics of those patients, would help us increase the probability of success for our drugs and for biomarkers. And so one of our big challenges has been incorporating the clinical research and the development sciences into our group. And this has had the main challenges, frankly, have been about process and about scaling up. So we've gone from being kind of a boutique operation to where we have to be doing things wholesale and doing things reproducibly, but with maintaining the flexibility so that when a new method comes along, we can actually incorporate it meaningfully and that everyone can have access to it. And there's a phrase that a colleague of mine uses, cattle not pets, where we have our data sets and until now we've sort of been treating them as pets and giving them names and being very solicitous of them. And at this point, we want to just herd them all into a pen and get what we can out of them. And that's a huge challenge from a bioinformatics science perspective. we're just for some details, we have roughly 60,000 patient samples in our freezers from trials that have gone back 10 to 15 years, and we're slowly marching through these and sequencing them, whole genome sequencing, and just scaling that up is a vast challenge. Not only the compute resources, which I think we're meeting pretty well, but where do you put the data? How do you make it accessible? Once you've done the primary analysis that you have planned, what then? The data are expensive enough that we don't want to just stash them somewhere. So that's something that, and we've been working with Bina to address some of these issues. Sort of along a parallel track, we have clinical trial data, so we have all the data that went into the filing for our various drugs and demographic data and detailed medical records for these patients. It's not that easy actually to get a hold of that. These are data that were collected for one purpose, and we're trying to repurpose them. And so the other big challenge is really figuring out how to integrate complex phenotype data with our patient data so that we can actually do systematic computation on them.
That's great. Thank you, Matt. So the next set of challenges around people challenges. What we are seeing is that there are some really big trends in the industry to create data-driven medicine, to make data really useful in discovering new biomarkers or drug targets and solutions. And the research has been always ongoing. So what is new today? It seems that next-generation sequencing has new challenges. For example, the volume of the data, the complexity, the scientific nature of it, that every day we see a new publication of how to make sense out of data around annotations or new bioinformatics tools. So it is a very dynamic field, and most of the organizations who are courageous enough to get into NGS domain, they need to build very strong teams that are capable of solving this problem, both from the infrastructure IT perspective all the way to the scientific and computational biology aspect of it. So my question to the panelists is that how do they find these people? And then also, these are large-scale projects. Usually you need many different expertise that many times is not in your team and are in other teams, either in your organization in different places or maybe across different organizations. How do they bring different people who might speak different languages even, they know different domains and they reside in different places. So maybe we talk a little bit about people challenges. Matt, do you want to go first? Because we were just talking about it before the panel.
Sure. So, we're really both blessed and cursed by our proximity to Silicon Valley, I think. There's a huge number of extremely bright, talented computer scientists, software engineers, data scientists, and we draw on these people for our putting together our teams. The challenge, I think, is that a lot of we're dealing with quote unquote big data, and that idea has gotten some kind of a mystique, right? And there are an awful lot of people who think they know how to handle big data, and they may, but the data we're dealing with is very, very different from the other kinds of big data that the Silicon Valley is dealing with. And we've had a number of cases where we've had consultants come in who are crack data scientists. And there's almost this image I get of, if we pour all the data in a bucket and shake it hard enough, science will come out. And we know that that's not how it works. The data is very highly structured, and more importantly, the way we handle it, the way we store it, has to be motivated by the scientific question that we have. And the frustration that we're facing is finding people who have this very, very strong quantitative background who also have a deep understanding of the science. And I think we're very lucky at Genentech that we've typically gotten people who are maybe biologists first who then learned the computation, or computational scientists who have always had an interest in the biology and always kept up with it. But it's not, you can't just plug and play computational scientists the way you can maybe with some of the hardware resources. There's and our other big challenge with talent is that there are now a lot of companies doing this kind of work, and so there's a lot of demand. So
Rong, do you want to also
Sure.
talk about it? Where do you find your team members and what are the challenges you see?
Yeah, so from my experience, we're more and more looking for people who are a good bioinformatician who also knows the sample quality, the DNA quality, the process that may cause these DNA problems, knows the database, can look at a BAM file saying this indel is correct or not, but also knows the scientific question and knows how to creating a report that help oncologists and physicians to say which drugs works better or not. So it's really becoming more and more requirement on the bioinformaticians that you need to start from the beginning, molecular biologies, to the raw, low-level qualities, also to the high-level clinical questions. And to answer your question, to finding the peoples, we continue having this problem, getting more qualified peoples. Just from a lab point of view, I found that LinkedIn actually works pretty well.
Good advice. Thank you, Ron.
Sure.
So the last set of challenges are around technology and IT infrastructure. We hear about cloud computing and the very specialized data centers large organizations are putting together to solve some of these challenges. And it seems that still not good enough in many cases, and people are looking for hybrid solutions that can run on premises for certain clinical applications or for cost effectiveness, and then use the cloud for elasticity and sometimes for centralization of the data and collaboration between different institutions. We see that genomics information is not helpful if it is isolated from other information. So how can you connect that genomic information to phenotypic information or other molecular information or imaging data is very important, and these systems usually are built by different interfaces or teams. And then we see that production grade, really end-to-end solutions are needed if we want to scale genomic research at institutes. So Matt, do you want to talk a little bit about how you are solving these problems at Genentech and where you see that there are biggest opportunities for progress?
One of the things that I learned very early on was not to optimize prematurely. It's like you have a problem, you need to solve the problem. Don't worry about how much time it's taking at first. Don't worry about how much memory it's taking. Don't worry about the resources. The first thing is to get the algorithm right. And I think a lot of us have taken that maybe a little, we've taken it appropriately to heart. But if you look at the tools we're using now, and this is true both for tools that we've developed internally and tools that we use that are commercial, that the premature optimization was avoided, but then the optimization actually has never really happened yet, right? So we rely on what are thought of as industry standard tools, but they're still kind of in this phase where they have not been made battle hardened for industrial scale, right? And so I think one of the insights that we've had in my group is we may be good at coming up with algorithms to solve problems. We're not software engineers, and we need to stop pretending we are, and we need to really outsource the expertise to companies that actually really know how to do this well. And I think that's sort of the trend that's happening in the industry that's most encouraging to me, is that we can kind of step back a little bit and we can prototype something and send it to a collaborator who's actually really good at developing the software, and trust that now we can scale up and get some real mileage out of that.
That's great. Thank you. Rong, we were just talking about how you think that industry needs actually to develop new database architectures and schema to help you with these complex annotation problems that you have. Do you want to talk a little bit about it?
Sure, yeah. So there are a lot of talks about new algorithms to kind of build index on the VCF files to allow you to query. But from my experience, the query we're really looking to solve questions are too complex to be handled by these simple index algorithms. The database is still probably the best solution to allow you to join hundreds of thousands of genomes with electromedical record to look at longevity, how it changes, and so on. However, so as many people know me, I already have at least 400 database, 150,000 genome in the database, but we're really hitting the bottleneck on the systems. We need a better a database system that can really handle like millions of genomes, allow you not just looking for a single variance across cohorts, we're really looking at the patterns that how these patterns being seen across hundreds of different disease cohorts and so on. And really looking at a single mutation, not like break this pattern, but creating new patterns. And this really needs more comprehensive, probably next generation hardware systems to really allow us to do more comprehensive query against this genome-phenome database. And especially if also you have an interface on top of this database, allow you to visualize all this trend of the data to answer questions. I think that can really help us along the way.
That's great. Thank you, Ron. So, to wrap it up, we heard that there are really different challenges in 4 categories: scientific challenges, technological challenges, people, and processes. At Bina, we want to build a product that solves all these problems. That might sound very ambitious, but what we figured out is that we cannot focus on only one of these challenges, because in order to really partner with the scientists, bioinformaticians, and clinicians, you need to have a one-stop solution that includes all of them. But at the same time, we realize that it takes a long time to develop this, and you need many different input and insight from the scientific domain, from the computational domain. So the way that we work with our customers usually is very, very collaborative, and we define the right set of features or capabilities that need to exist in this product to solve both analytics problem, data management, as well as the kind of people framework to bring different expertise together. So I want to open up the floor for questions to our panelists. Also, I want to remind everyone that we have a poll going on. You can have your voice heard around, what are the bioinformatics challenges if you go to bina.com/poll. So I have some questions for our panelists, but first I wanted to open up to the audience to see if there is any questions. There's a microphone in the front if there's any questions. Okay, seems everyone is very content. So I will ask. Okay, go ahead, please.
Hi, I was just curious about how companies like Genentech are thinking about handling, okay, so there's genetic variation, right? But there's also epigenetic variation and genome organization and all of these different things that are layers and layers. Different tissues have all of these different properties. We're having challenges just with the genetic part, let alone layering all of that stuff on top of it. So I'm just curious how big companies are thinking about handling that.
I think it really comes down to the scientific question that you have. So for our non-oncology programs, the genetics is sort of the primary question that we're asking. We're not taking tissue-specific samples in any way. On the oncology side, on the other hand, you need tumor samples, and preferably matched normal, and there's something to be gained from RNA-seq, for example, from tumor normal. I think the tendency is to want to get comprehensive data sets for everything, and the reality is there's just not enough resources for that. So, if there's a good indication that there's an epigenetic component to some disease, then we'll focus on getting those samples and getting that data. But we really do want to avoid just sort of covering the whole universe of possibilities, because it's just we have enough to deal with with very well thought out primary questions as it is.
One step at a time. Thank you, Matt. So I have a question for our panelists. I know there are several vendors also in this room, and they're all working towards solving the informatics challenges of next generation sequencing. And sometimes for startups and companies, it's hard to know where they can add value, where they can be most impactful, what are the gaps that they can go and make a difference. So if you wanted to give advice to the informatics companies, where do you think they should focus to be most useful to your research?
So for me, I think the biggest value is still in the biobank. My dream has always been 1 million genomes with 1 million people's electromedical record with a longitudinal clinical measurements, the clinical notes, all these. So I guess from a company point of view, if you have a product using either insurance pay or self-pay to support you to really scale up to get to this 1 million genomes biobank. That will be the biggest value.
Thank you, Matt.
I guess it's not quite as a grand vision, but we need help in bringing our ideas into sort of a production system, right? So I think there are a lot of vendors I talk to who have great ideas, but who have not really listened to our scientific needs. And we have a very specific algorithm we want to implement. We'd like to have that algorithm optimized. We don't want their algorithm, even though it might be very good, optimized, right? So I think getting in close with the scientists you're supporting and listening to what the scientific use cases are and the scientific needs are, and understanding that the bioinformatics scientists are not software engineers, but they're still highly computational, and they're not going to want to necessarily give up all of the power about with the data.
Makes sense. Thanks so much. We have some questions, Matt.
Last month, the NIH had an RFI out asking the question of where do you see a need for exascale computing coming in biomedicine? I've talked to a number of people who believe that there is an order of magnitude or more to be gained merely by algorithm optimization. But I'd like to pose the question to you, where do you see the need for computing on a scale 100-fold bigger than we do now?
So yes, we are replying, submitted to this RFI at Mount Sinai. So currently, like I said, we have these 100,000 genomes, but in our database, we can only looking at single variants. We are not able to look at the patterns, combination of variants across all these hundreds of disease cohorts. So if there is I believe the database is still the best infrastructure to kind of handle these complex, open-ended questions, but the current database infrastructure are not able to hosting these million, at least 200,000 whole genome coming out in the next 4 years, right? So how we have a database systems that can hosting all these complex phenotypic information and allow you to query not just for single variant, but for combination and patterns, and also has an easy interface, allow you to run statistics and to visualize, explore all this phenome-genome relationship. I think that will be great.
Great. Okay, go ahead. Next
Thank
question.
you. So one thing I haven't heard any talk about yet is compression of the data. I'm from the VC world actually, but one of our portfolio companies is working on this problem. FastQ, BAM, already achieving double-digit compression ratios. And I'm wondering, what do you think are the biggest opportunities and challenges with compression specifically?
So, if I could take that. So when we started on our program to get whole genome sequencing data from our clinical trial subjects, the initial idea was we couldn't afford to keep the BAMs. We were going to keep just the VCFs. And that would have addressed our primary questions very nicely, to have the GVCFs for the patients. But obviously, there's a huge amount of data on genome rearrangements and structural variation in the BAMs. Right now, we are sort of winging it in terms of keeping the BAMs. We're hoping that we can grow our storage fast enough that it's not going to be a big problem. But I'd probably like to talk to you after this, actually. Yeah.
Great. Thanks everyone for joining us for this panel. Thanks to our panelists.